This has been an exciting meeting. I think for me, so far, the two main highlights really were the lectures by Andrea Califano and Jim Allison. And although they were quite different, I think they illustrate a very important principle. So, Dr. Califano's lecture, I think, highlighted the enormous complexity of cancer and the fact that it may be possible through very careful and innovative analysis of molecular networks to identify key nodal points where there are master regulators of those regulatory pathways that, if targeted, could potentially have a major impact on the treatment of cancer. But what he really illustrated was just how complicated each cancer can be. Now, Dr. Allison, I think had the perfect response to that, which is that the only thing more complicated than cancer is the immune system. And the ability of cancer to generate heterogeneous subclones can easily be matched by the ability of the immune system to generate enormously heterogeneous responses. And so we're beginning to see now the enormous promise of immune therapy in treating cancer, and the fact that it seems to be able to be effective against essentially every kind of cancer because it's agnostic with respect to things like histological type or mutational status. So these two concepts about the heterogeneity of cancer and how to understand it and target it, and the capacity of the immune system to match that heterogeneity and then, kill the cancer in a tissue agnostic way, I think are really two of the real highlights of the meeting. Yeah, well, I think one of the keys there that his lecture and others, other researchers are working on are, signatures of immune response that can predict which cancers are more or less likely to be susceptible to attack by the immune system and by which components of the immune system. And so, because of the great expense associated with modern cancer therapies, it's becoming more important than ever before to be able to identify the patients most likely to benefit from any particular kind of treatment, because to say that we can afford to just treat everybody is unrealistic. It's just no healthcare system is going to be able to sustain that. So we have to really be able to understand the biology well enough to develop the biomarkers to understand which patients require which therapies, and then focus those therapies on those people in particular. We talked in that session in the roundtable primarily about strategies to overcome drug resistance. I don't know that we drew any major conclusions from that, except to say that combining drugs is complicated. It's not only in terms of selecting which drugs to combine, but how to combine them, how to derive the doses of the drugs that can be given safely in combination, how to sequence the drugs. Is it necessary to give 3 drugs together? Can you give them sequentially in some way? How to determine a biomarker for the impact of a drug combination, on a patient's tumor. So I think all of these are points that came out during that discussion. We talked a little bit about also, combining small molecules with immunotherapies. I think people all see some value in doing that, but again, it's a question of how to do it safely. So I think that was really the focus of that conversation. Then in the second part of that session, we really began to get into a review of a WIN portfolio of clinical trials. So we had an update on the status of the WINTHER trial. And that, of course, was the first of the WIN trials. And one of the novel things about WIN is that it's, in a sense, been willing to go where no other trial organization has been willing to go, which is to go immediately to combination therapies in very difficult to treat cancer and to approach it in a very innovative way and to engage a representative group of institutions from around the globe to tackle the problem. Well, there are none being conducted yet, but there are 3 being planned, all in lung cancer. One is sort of a tissue acquisition platform study called BOOSTER, which is a study designed to identify biomarkers of people who are likely to have or develop lung cancer. So that's a study in which patients who are newly diagnosed with lung cancer will have pre-treatment biospecimens obtained, both blood and serum and tissue, and all the biomarkers are yet to be decided. Then we'll have definitive surgical resection of their cancer. The tumor will be obtained, the normal lung tissue will be obtained, and then we'll have, in the postoperative period, sequential acquisition of additional biospecimens. And with that, and of course we'll capture the long-term clinical outcomes from these patients, so with that we'll have a very rich data set of biomarkers and clinical outcomes that can be used to identify those biomarkers that might actually be useful in either identifying a cancer very early on or monitoring for recurrence. Then you've got the SUMMER trial, which is really an adjuvant postoperative therapy trial for patients who have a resectable lung cancer and for whom adjuvant postoperative treatment is a standard of care. And the idea of SUMMER is to randomize patients between a standard of care treatment, presumably chemotherapy, versus chemotherapy with one or more targeted agents, with the targeted agents to be selected based upon the SIMS algorithm for identifying targeted treatments. And then the third trial, the SPRING trial, is for patients with metastatic non-small cell lung cancer. I think that trial has been the most controversial so far with respect to its design. But the ultimate goal of SPRING is to improve the 5-year survival rates for patients with non-small cell lung cancer by giving 3 targeted drugs in combination. Now, the hard part is to figure out which 3 drugs to use because the 3 drugs might be different for different patients because of this whole issue of cancer heterogeneity. And so, and that's where things like the SIMS algorithm, which allows one to interrogate a cancer both with respect to its mutational profile and its transcriptional profile, and then integrate that information using the algorithm to identify the drugs most likely to be effective. That's what needs to be tested out in the SPRING trial. I think the challenges are several-fold. So, every country in the world has a regulatory authority, a health authority that regulates whether new products can enter the medical marketplace. And the new products, can take the form of drugs, or they can take the form of devices or tests or, other types of medical products that, are used either as machines or prostheses or whatever. So, the regulations that apply to authorizing drugs for marketing are different from the regulations that apply for tests, entry into the marketplace. And of course, different jurisdictions of the world have different laws and different regulations. So if you're trying to do global clinical trials, like we're trying to do in WIN, where you have countries all over the world participating, obviously, the ability of each country to launch a trial depends upon its regulatory authorities, and it has to be able to comply with its local laws and regulations. So to the extent that these can be harmonized to the greatest extent possible, it would greatly facilitate the launch of global clinical trials. Otherwise, you find that one country can launch, another country can't launch until 2 years later. So I think the goal of the roundtable is to have a conversation between the academic clinical trialists and the regulatory authorities about what are the challenges, what are the goals, what are the obstacles to achieving some degree of harmonization on regulatory principles that will allow these global trials to launch in a much more comprehensive way across multiple countries, multiple jurisdictions, all at the same time. I think the WIN Symposium program this year has been fabulous. It's been forward-looking. It's been creative. We've had wonderful speakers. And I think it's, the symposium, in a sense, is the most visible part of WIN, but a lot of the most exciting parts of WIN come from the engagement of all the WIN participating institutions and the development of what I think are a very innovative suite of clinical trials.